So we've got these engineered bacteria that actually like target tumors from the inside out by exploiting the oxygen-starved core.
Mm-hmm.
It's pretty wild, right?
Yeah, that's amazing. These bacteria, like Clostridium sporogenes, naturally thrive in no oxygen conditions, making the tumor core their perfect home base, correct?
Exactly. It's like they're essentially turning the tumor's own environment against itself. When the bacteria find that nutrient rich, oxygen poor zone, they start multiplying and devouring the available resources inside the tumor.
Right. But what about when these bacteria hit the tumor's outer edges, where there's a little oxygen? How do they manage to survive there?
That's where it gets really interesting. Researchers added a genetic tweak, a gene from a more oxygen tolerant bacterium that kicks in only when there's enough bacterial density through quorum sensing. So it's like the bacteria wait until they're packed enough together to
Yes.
signal the switch.
So essentially, the quorum sensing mechanism ensures they don't prematurely activate the oxygen resistance, preventing them from causing issues in healthier tissues like the bloodstream, right?
Exactly, you got it. It's all precisely timed. The system only flips when the bacteria have sufficiently colonized the tumor core. It's like assembling an army and waiting until you're strong enough before moving outwards.
Mm-hmm. Now, I wonder, don't you think there's some risk with using a living organism in such a delicate scenario? I mean, can we fully control how far they spread?
That's a great point. The design purposely delays the oxygen resistance gene activation until the bacterial population reaches a threshold. This keeps them contained initially in the tumor. Sure, there's always a precaution factor, but the programming via synthetic biology and DNA circuits really minimizes those risks.
Yeah, and it's really neat how they use a natural bacterial communication process for control. It reminds me a bit of how social media trends spread. They say when everyone's on board, then things really take off.
Oh, for sure. It's like using the bacteria's own group chat to decide when to, level up their defense mechanism. It's elegantly engineered using components that act almost like an electrical circuit, but with DNA.
Right. And this whole interdisciplinary approach, with experts from chemical engineering to applied mathematics and microbiology, really showcases how complex challenges like cancer require collaborative innovation.
Exactly. This project, involving people like Dr. O'Coin and Dr. Ingalls, along with dedicated PhD students, is a shining example of how bringing together diverse expertise can lead to completely new therapeutic strategies.
Mhm. I do wonder, though, could there be any unintended side effects from the genetic modifications? I mean, nature's always unpredictable, right?
True, and that's a fair concern, but the rigorous preclinical trials planned aim to address just that by carefully monitoring these engineered bacteria in controlled settings. It's a promising frontier in cancer therapy, blending synthetic biology with medical innovation. It's
Yep.
an exciting time for this research.